Hello everyone. So in this video we're going to talk about enzyme catalysis and its related terms. So let's start with the definition of enzymes. What are enzymes? Enzymes are highly efficient and specific catalysts. So enzymes are proteins. Some of the enzymes are RNAs too that are known as riboenzymes, or we are talking about general enzymes. So these are mostly protein molecules that act as biological catalysts. Enzymes are biological catalysts or biocatalysts that accelerate the chemical reaction. So what is saying is that enzymes are protein molecules that act as biocatalyst and which accelerate reaction rates within the biological system or within cells. Right. So the molecules upon which enzymes may act are called substrate. And the enzyme converts these substrates into different molecules known as products. So what happens if this is an enzyme, it binds to specific substrate, and within this substrate old covalent bonds are broken and new covalent bonds are formed, and thus this substrate is converted into a product molecule. So substrate is converted into product. This is the main function of the enzyme, converting the substrate into products with a high reaction rate. The reaction rate is increased. Now thousands of different type of enzymes, each of which catalyzes a single chemical reaction or a set of closely related reactions, have been identified. Which means what? That, like I said earlier, enzymes are highly specific catalysts. That each enzyme only act as catalyst to only specific kind of reactions, unlike chemical catalysts which may act as for catalyzing many kind of reactions. Enzymes are on the other hand are very specific. So this is a cartoon and it shows how the enzymes actually function. So for example, if hydrogen peroxide, which is H2O2, is the substrate that is entering into the active site of the enzyme, which is known as peroxidase enzyme. So peroxide substrate is entering into the active site of the peroxidase enzyme, and what is happening is that enzyme substrate complex has been formed. Enzyme is now bound to the substrate and formed a complex. In the next step, what happens? The bond between these substrate are broken and new bonds are formed within these substrate, and thus this substrate is now converted into these products. So now enzyme product complex has been formed, and these products are what? Water and oxygen. So H2O2, the hydrogen peroxide, is now converted to H2O and O2, which is water and oxygen, by these enzyme known as peroxidase. So let's see how this actually happens, right? So the enzyme is binding to substrate and converting them into product. This is clear. But how and why this is happening? Why the reaction rate are increased if enzyme binds to the substrate? So like all catalyst, enzyme increases the reaction rate by lowering the activation energy. So what is this activation energy? Let's see. So for a reaction to occur, or to happen, what we need is the substrate needs to reach a transition state. And the energy that is required for this substrate to reach this transition state is known as activation energy. So for any reaction to occur, that substrate needs to reach a transition state at higher energy level, and the energy that the substrate needs to reach this transition state is known as activation energy, right? So every substrate needs to have this activation energy for the reaction to occur, for the products to form, right? So let's see. So what is happening is that in general, when there is no catalyst, what is happening is that these are the reactant at this low energy state. The reactants are here, and so it needs to reach somewhere here, the transition state. It needs this much energy, or activation energy to reach this state. Only after that, after reaching this transition state, can this form product. So every reactant, for the reaction to occur, requires this much energy to reach this transition state, and only then the products could be formed. So what the catalyst actually do is that the catalyst actually reduces the transition state, or reduces or lowers the activation energy. So here you can see, when catalysts are present, what is happening is that transition state is lowered down from here to here. So when there is a catalyst present, the reactants only reach to have this much activation energy and not this much. Only this much activation energy is now needed for it to reach a new transition state which can form the product easily. So thus, this is how the catalyst actually increases the reaction rate, by lowering down the activation energy. Instead of this transition state, on the uncatalyzed state, now it could convert to product only by having this much energy, because the activation energy has been lowered down by the enzyme. So here is another cartoon to explain it very easily. So what is happening here is that enzyme and substrate are present. When there is no enzyme, when there is no catalyst or enzyme, it needs to have this much energy. Here is the transition state without no catalyst. But when enzyme or catalyst is present, what it needs to do is only reach this much energy, or this transition state, with the bound substrate. Enzyme substrate complex has been formed at the transition state, and now it can form the product. So instead of this higher transition state, now because the enzyme is present, only this much energy, this much activation energy is enough to convert the substrate into product. So enzyme increases the reaction rate by lowering the energy of transition state, and therefore the activation energy required to reach it. Right. So now what we next need to study is about the activation site or active site that binds to substrate of an enzyme. So an enzyme's active site binds substrate and carries out catalysis. So an active site of an enzyme consists of two functionally important regions: the substrate binding site, which recognizes and binds the substrate, and the catalytic site, which carries out the chemical reaction once the substrate has been bound. So like you can see in this picture, this is an enzyme. What is happening here is that there is a substrate binding site. This is complete active site, and the active site has one substrate binding site and one catalytic site. So at the substrate binding site, the substrate binds to this site, while the catalytic site catalyzes the reaction after the substrate has been bound. So this constitutes the active site of an enzyme. So mechanism of enzyme activity. So here you can see how the mechanism of enzyme activity occurs. So this is a substrate, and this is the enzyme, and this is its active site where the substrate binds. And so now the substrate is now bound to the enzyme. As you can see, substrate is now bound to the active site of the enzyme. An enzyme substrate complex has been formed, which goes to the transition state. And this enzyme substrate complex, now these bonds between the substrate are broken and new bonds are formed. Bonds broken and new bonds are formed. Thus, products are now formed. Substrate is converted into products, and these products are released from the enzyme active site, and the enzyme is once again ready to bind to new substrate. So here is an example with names. So if this is a substrate, which is sucrose is made up of glucose and fructose. So this is a sucrose molecule, and it's bind to an enzyme together to its active site, And so the substrate binds to the enzyme, forming an enzyme substrate complex. Here enzyme substrate complex has been formed, where the substrate is bound to the active site of the enzyme, and now dehydration is occurring, a reaction is occurring, and the bond is broken between them and new bonds are formed, and thus glucose and fructose are released as products. So products are released and the enzyme is free to bind to other substrate. Now this is also known as lock and key model. Lock key model of the enzyme activity. So the idea that substrate might bind to enzyme in the manner of key fitting into a lock was suggested by M.L. Fisher in 1894. So what they suggested is that the substrate acts as a key, right? Like the key fits into the lock and is very specific that only the key to that lock will fit into that that active site, is how this is shown. For example, if this is the key and this is the lock, enzyme is the lock, and the key or the substrate fits into the active site of the lock or the enzyme, and thus only then the reaction could occur. So this is known as the lock and key model of enzyme activity. Here another cartoon is showing the lock and key method, lock and key model given by M.L. Fisher, lock and key model. So you can see the substrate is exact fit to the enzyme active site. This is conical and this is conical, exact fit, like a key fits into a lock. So the substrate acts as a key and it fits into the active site of the enzyme, and this enzyme substrate complex is formed, and then catalysis occurs, old bonds are broken, new bonds are formed, and there's a product is formed, and enzyme product complex is formed, and product are then released, and enzyme is once again ready to bind to another substrate. So this is lock and key model, where the substrate acts as a key that fits exactly into the active site of the enzyme. Now the second model is induced fit model. Induced fit model after lock and key method, which says that enzymes are flexible structures. Active site can change the shape to fit with substrate, and which is better and widely accepted theory. So what it says is that, for example, if this is the enzyme and this is its active site, which you can see does not fit the substrate, so what is happening is that it is flexible, the active site is flexible and changes the shape according to the substrate. It is changing its shape. It was conical first, like something like M, but since the substrate is round, it is changing its shape of the active site and becoming round, and then fits with the substrate, and thus enzyme substrate complex is formed. So this is a different model than the lock and key model. This is induced fit, that at binding with the substrate, the active site also of the enzyme also changes. So Daniel Koshland in 1958 called induced fit model. He gave this induced fit model, which said that the substrate binding site is not rigid as a lock, but is flexible and is induced to change shape for more optimal catalysis when substrate binds. Right? So we say that unlike lock and key model, what this induced fit model says is that it's not the active site is flexible and not rigid, that it can change its shape according to the substrate for a better catalysis. So this is induced fit model. So thank you for watching. Please like and subscribe. In the next video we talk about the Michaelis-Menten equation and its graphs. Thank you.